specifically in the spider silk series. So we're right here. We're still in the area of protein folding, silk folding, but also today I would like to show you now how to make proteins, how to use biotechnology to get single proteins. So these will be the topics of today. So first, in the first part, we will still come back to the protein folding issue, and as I said yesterday, I will give you some more details today. I will talk a little bit about non-covalent interactions, because these are the most important interactions that we need for assembly. So when we get proteins together, and of course for silk that's a big issue, because we have millions of proteins in a fiber, so they have to interact, and they typically do that in a non-covalent manner. And as sort of like a special treatment, I will give you a very brief insight into the structure of water. The reason is that silk fibers have, well, quite a high ability of absorbing water. And even if you have a dry silk fiber, there is still about 6% weight per volume water incorporated. So we call that structural water or internal water, because water serves as a plastifier for the structure. You only can remove the water out of a silk fiber if you put the fiber into vacuum or even high vacuum, and then also you lose all the mechanical properties. So water is very important for the structure, and water has a structure itself, so therefore I would talk about that. So that would be then the end of folding and assembly and interactions, and then I will make a break and then start with something completely new, and I will talk about how to process proteins, how to make proteins. As I told you yesterday very briefly, it's very important if we want to analyze and understand folding and assembly of proteins, especially of silk proteins, we need a homogeneous system. So we need single proteins to work with, and therefore I will give you an explanation how to do that. Since probably you don't have that much of background in biology, I will give a very brief glance on what we call transcription and translation. So this is how proteins are made in nature. And then I will give you insights into how to recombinantly produce spider silk proteins. What does recombinant production mean? Well, we choose a host organism that we can manipulate. In our case, it will be bacteria, for instance. And we actually introduce the genetic information of a spider into such bacterium, and that bacterium will then produce silk proteins, and this is what we call recombinant production, and this will be the final part of today's lecture. To get started with, I mean, this is something we had already yesterday. Why do proteins adopt a defined structure? We talked about protein folding, and then also we have to talk a little bit about entropy. I mean, yesterday we heard lots about thermodynamics and a little bit about kinetics. Today I would like to stress a little bit the term entropy. If we see the unfolded state of a polypeptide, that was the U yesterday, we see that the unfolded state of the polypeptide has, of course, a higher conformational flexibility than the folded state. If you have an unfolded chain, it can tangle much more than if it's folded. But still, I told you that the folded or the native state is not a stiff structure. It still is flexible, but it's much less flexible than the unfolded state. What does this mean? Well, if a protein folds from the unfolded to the folded state, it loses entropy. This is very important. It loses entropy upon folding. If we now think about how such folding takes place, we have to consider the amino acid composition. So that's what we called yesterday the primary structure, right? So the primary structure directs folding, and this is due to the case that amino acid side chains can interact with each other. For instance, they can form HH bonds, and we will talk about this in a minute. These interactions actually mean that amino acids now non-covalently interact and form sort of, well, contacts that also lead to loss of entropy upon unfolding. So a second loss of entropy in addition to the first one. So then we can have some chemical stabilization in some cases. We can have disulfide bridges, or we can have the proline residues, and we talked about the proline residues yesterday. Because the side chain of the proline is a ring structure, it's a structure, it's especially a helix breaker, but it also interferes with better sheet formation. So if we have proline residues and also disulfide bridges involved, but disulfide bridges play less a role in silk proteins. So if we talk about the proline residues, we also lowering the conformational flexibility, and this, on the other hand, will stabilize the folded state. If we have a look at the loss of entropy, then it antagonizes folding, and this effect increases with increasing temperature. So entropy changes are always temperature related, meaning that at high temperature, proteins unfold. And you know that boiling of an egg is a typical example for that. So you have the proteins in the egg. If you just put that to a hot pan, immediately the proteins will unfold and then they will aggregate. And this is what you see by gaining the white color. These are protein aggregates. And proteins can only aggregate once they're unfolded. So therefore, this is the simplest picture you can imagine about high temperature unfolding of proteins, boiling of an egg. So an important point, of course, is also the hydration shell, so the water that is surrounding the polymers. We talked yesterday already a little bit about the hydrophobic collapse model. So we do have amino acids with hydrophobic side chains. And of course, hydrophobic side chains try to avoid contact with a solvent, especially with water. And therefore, hydrophobic side chains typically are buried inside a protein structure during folding. What does this mean? Now, if we look at the solvent, this is a gain of entropy because the solvent does not longer have to deal with hydrophobic side chains, which can interfere with the structure of water, as we see later. So therefore, we have here a gain of entropy in the solvent. So we have loss of entropy concerning the molecule. We have a gain of entropy concerning the solvent. And this is one of the driving forces why proteins adopt a defined structure. And this is what we have to keep in mind, again, if we talk about spider silk. Of course, if we check now all the atoms of a protein, then we will find that they can interact differently in the folded state in comparison to the unfolded state. So this is what we call the enthalpy. The change of enthalpy is lower than 0. And of course, the positioning of each atom is accompanied with a loss in entropy. So the change in entropy is smaller than 0. And we can actually bring this all together by the term here. So this is the free energy that we find in the system, and this is actually derived from the change of enthalpy and the change of entropy, and the later is, of course, dependent on temperature. So this is actually the major term of protein stability, and this is actually the reason why proteins do adopt a structure. So the proteins fold, and therefore the side chains and the atoms have to interact, and this is the list of non-covalent interactions that we do find in a protein. They play a central role, and they're really important concerning protein stability, but also concerning binding of ligands. So if you think about an enzyme that binds a ligand, then we have non-covalent interactions, and of course, they're important in assembly. This is what we find in fiber assembly, for instance, in spider silk. So let's go through that very, very carefully. So the first interaction is called van der Waals interaction. Which atoms can form van der Waals interactions? Well, all atoms can do that. There is no requirement. However, the energy that is provided by such an interaction is quite low. We have less than 1 kilojoule per mole. So van der Waals interactions occur everywhere in the protein. Their contribution to stability is low. However, the sum of all these very low energy impacts can be quite sufficient for stabilizing a structure. Then we have electrostatic interactions. Electrostatic interactions occur with amino acid residues that do have charged side chains. So we have positively charged amino acids, as I told you yesterday, and positively charged ones, and they can make so-called salt bridges. The salt bridges can occur on the surface, and as you can see on a surface, the energy input is not very high. It's around 4 kilojoule per mole, and this can be explained by the exposure of the salt bridge on the surface. Of course, there are interactions then with salts that surround the media, that surround the protein in a medium, and therefore the energy input is much smaller. Salt bridges inside a structure have much higher energy input and stabilize the structure much more efficiently. However, if we check where are electrostatic interactions, they're typically on the surface. And this is based on the fact that charged amino acids are typically hydrophilic, so they're pointing towards the solvent rather than pointing inwards, because pointing inwards is typically something that hydrophobic residues do. Of course, what we need is charged side chains, and of course, these electrostatic interactions are pH dependent. And we saw yesterday already an example. The clamp in the C-terminus of the silk showed a pH dependent structural change. And this is based on the salt bridges that actually hold these two proteins in position. And once we change the pH, they can change the conformation. So H bridges are also very important, especially for silks. And you see that we have here quite some energy input from a single HH bond. It can be intramolecular, meaning it can be an H bridge between amino acids, but H bridges can also form with the solvent. And this is where water comes into play. And this is why water is also very important for stabilizing a structure. It can form H bridges with amino acid side chains. And therefore, you get, so to say, an interaction between a single water molecule and your amino acid. And this can stabilize the structure quite dramatically. So what you need is then an H donor and an H acceptor, but in amino acids that is typically no big deal because we have both provided in a protein. And the final of these four non-covalent interactions are hydrophobic interactions. Also, the energy input is quite high, 20 to 50 kilojoule per mole, depending on the environment in which such a hydrophobic interaction takes place. It's typically inside a structure because hydrophobic residues are not exposed on the surface. So, and of course, as a requirement, you need hydrophobic amino acids. So let's have a look in more detail. Let's start with the electrostatic interactions, as I showed you yesterday. So you need a pair of charged amino acids, and this would be a pair coming especially from silk. So we have a glutamate, negatively charged, and we have an arginine, positively charged, and they can form what we call a salt bridge. So charged side chains, or also dipoles, are usually surface exposed. In terms of the silk protein, that's also the case. If you remember the structure, these were these two helices, and the salt bridges were located here. So they are typically on the surface of a structure. It's very important to know that there's no directional dependence. So it doesn't matter if the glutamate is turned in that direction or the other way around. This really doesn't matter. You see here sort of a symmetry, so therefore you can turn it around in any direction, and the same is true here for the arginine. Important is also that it's a long-range interaction. So the amino acids can be far apart, and they still can interact because you have a play of attraction and repulsion that works, and this works over larger areas. When we then investigate the van der Waals interactions, you will see that these are only short-range interactions. So important is also that ions play a role here, pH plays a role, and also the solvent plays a role, and I don't want to go into much detail here. So electrostatic interactions can not only occur between charged amino acid residues, it can also occur if you just check the peptide bond. So this is just a different drawing what we saw yesterday from a peptide bond. And what you can see here is that a peptide bond itself is, so to say, a micro dipole. Why is that so? We have differently charged side chains. We have an asymmetric charge distribution along a polypeptide backbone, and therefore we have here a system that can function as a microdipole and that can also make charge-charge interactions. And we make use of that when we make beta sheet structures. So there the beta sheets can form just by packing the peptide bonds in a manner that these microdipoles actually interact electrostatically with each other. Also, p-electron systems, like in aromatic amino acids, can show these dipole character, and they can also form very tight stacking. So this is a different type of electrostatic interaction in addition to the charged amino acids, but this is also of importance for protein stability. So this is now the contrary. As I told you, charged interactions are typically long-range interactions. Van der Waals interactions are typically low-range interactions. And here you see why. Van der Waals interactions typically occur between two atoms. And if we just bring two atoms close to each other, there is two different ways of action. One is attraction of the atoms and, of course, of the electron shells. And the other one is repulsion. And you see here that if you just sum up attraction and repulsion, in red you see what van der Waals found out, that there is a minimal energy or an energy optimum at the optimal van der Waals distance. So meaning that here at this point you will have a van der Waals interaction holding the two atoms in position, because this here is a stable state. If you move the atoms farther off, they will just repulse and move away. If you come closer, of course, repulsion will get stronger and stronger, and you can't push them, because then the atoms would have to melt, and this is an energy that cannot be overcome. So it's actually a very, precise interaction based on precise packing of individual atoms inside the protein. So it's highly specific. The positive thing is it can occur between all types of atoms. So every atom in a structure can actually contribute to van der Waals forces, but the packing is important, it's very specific, and the distance between the two atoms has to be optimized that you have van der Waals interactions. H-bridges. As I said before in the table, for making an H-bridge, you need an H donor and an H acceptor. So the hydrogen is actually shared between the donor and the acceptor atom. This can be the case, for instance, for two electronegative atoms, let's say nitrogen or oxygen or sulfur, and they share one hydrogen atom. Interestingly, the distance between the hydrogen and the other atom is smaller than the sum of the van der Waals radii. And this makes the HH bond energetically so strong, and therefore H bridges are very important in stabilizing protein structure. So we do have an electron delocalization, so there's also electrostatic aspects involved in the H bond formation. And of course, we have there an independence of direction and distance. Again, it doesn't matter in which direction the atoms point, because the interaction can take place anyhow. And the distance, again, is more a long-range interaction. If we check HH bonds in peptides, we typically find them in the peptide backbone, and therefore the H bonds are the main driving force for the determination of secondary structure. So actually the way how H bonds form determines if you get a helix or you get a beta sheet. So this would be actually the major interaction that we have to manipulate if we would like to influence the structure of our final silk fiber when we're looking for the materials. Because the sheet is more stable than the helix, and for some materials this is very important that we control the structure. So it will be important to control the formation of H bonds, and this is particularly of interest because H bonds can also compete with water. Of course you can form H bonds between the donor and a water molecule. This means that the acceptor can be water, and we can have here competition in the structure, and therefore we have to have a very close look at what the role of water is and how water interacts with the peptides. So the final non-covalent interaction is the so-called hydrophobic effect. What is actually the basis of this effect? It's a simple observation. Non-polar molecules aggregate if you put them in polar solvents. So that's simple chemistry. And the explanation for that is that around such a molecule, the formation of an ordered shell, especially a hydrate shell, is energetically disfavored. You have something that dislikes the interaction with water, that does not make H bonds. Therefore, the water does not like to make a structure surrounding this molecule. As you see here, it has to form sort of a cage, and this is entropically disfavored. So therefore, such a cage has to be avoided by the system. So therefore, hydrophobic side chains typically pack together to lower the surface area to the contributing and to the environmental water. And this is the reason why we have hydrophobic amino acid residues typically inside of proteins, because then you can avoid the contact to the solvent. We have a very low specificity. I told you already the example of boiling an egg. You can see that if you just unfold the egg case proteins, the egg proteins, you just get spontaneous protein aggregation. The reason is that the hydrophobic amino acids are not now exposed, and now they can interact with other exposed hydrophobic amino acids, and this is what we call protein aggregation. So we have a low specificity. They don't care about their partners. I mean, as long as they are hydrophobic, they will interact. And this, of course, is an unwanted side reaction of assembly. Again, if we think about silk proteins, we have to avoid protein aggregation, right? We want to make fibers, aligned structures with a very high precision. Protein aggregates have low precision, no order. So therefore, this has to be avoided. And this is one of the reasons why we do not find extensive hydrophobic amino acids in spider silk sequences. If we check these core sequences, we don't see a lot of aromatic amino acids, because they could cause unspecific protein aggregation. So this was now just a very brief insight into non-covalent interactions. And now another insight will come to the solvent. Of course, the solvent of a protein will have influence on the protein structure. If you have a protein, or in general terms, if you have a polymer in a good solvent, it expands its chains, meaning that you have no structure. The interactions between the solvent and the side chain are favored, and therefore you should have an unfolded molecule. What actually is then the outcome? That in a good solvent, the protein structure should be random coil. But in nature, that's typically not the case. So what we have is we have a folded structure, so a contraction. This means a bad solvent induces the contraction of a polymer of a protein, to minimize the interactions to the solvent. As a result, we see water is a very poor solvent for most proteins. And also, there is not much internal water in a protein structure. And now we have one exception, this is silk. Because silk is unfolded in water, besides these terminal domains, but the rest has no structure in water. And we do have a lot of internal water once we make the fiber. So actually, water is quite a good solvent for silk molecules, but water is a bad solvent for most of the other proteins. This is very important, and this also will be important then if we talk about processing of silks, because we can use that issue quite a lot, and we can actually manipulate the proteins much better because they are very well solved in water. So this is really a very exceptional thing for silk proteins. They behave not like a protein, they rather behave like a polymer in a good solvent. And this is very untypical. And this is what most people haven't, well, taken care of in the past. But you can use that topic and that issue, and this is very exceptional. As I told you, a few words about water. You know water is H2O, of course. If you have a look at the structure of water, and you know that also, I'm sure by that, water is not a linear structure. It has here an angle. So the two hydrogen bonds, or the two hydrogen atoms, are separated by 104 something degree. Meaning that we have here a dipole moment. So we have here partially positively charged hydrogens, and we have a partially negatively charged oxygen here. This is very important because now this molecule with the negative charges here and with the positive charges here can interact with each other. This means water can form H bonds with itself, and this means that water can form structures. So as you can see here, we can actually bring water in this pyramidal shape, and it can even go on. Water has very strong interactions and it's forming really very tight structures. So here you see a bicyclooctamer, and you see even more. So it can form cyclic pentamers, bicyclooctamers, tricyclodecamers, and so on. So this is what we call water clusters. So if we talk about water, actually we talk about a structured chemical. And you can see that water has a very precise structure, but by many different effects. We call that a normal behavior of water. You know that the density of ice is higher than that of water, for instance, boiling temperature, and so on and so on. There's 38 abnormal features that you find in water that are actually dependent on these water clusters, on the structure. This is on a low scale. You can also see that on even larger scales, and you see how these clusters form, and this is a dense network of structures. And now you can imagine why water is, for most proteins, a bad solvent, because it's entropically not favored to disrupt now the structure and bring here anything inside. It will destroy the structure, and this is not favored. And these structures are, again, highly dynamic, and you can see here these icosahedral clusters. They can actually somewhat breathe. You can have like open structures with low density or condensed structures. So it's like pulsing structures. They can move. They're highly dynamic. And this is very important now to understand, because this is what we call bulk water. Bulk water has structure. And now we put in their proteins and we destroy this structure. And this is energetically not favored. So if you would like to bring something in, it has to integrate into the network of water very well, and then water will act as a good solvent. And this is what we find in silks. And therefore, we have to pay a lot of attention to this water structure and how water behaves if we would like to understand protein structure. So far we have talked about gaining structure. So we have an unfolded polypeptide, and we try to structure it. Now I would like to introduce briefly how to unfold a protein. Why is that of importance? Well, in the past, when people tried to study silk molecules, what they had were fibers created by the animals, the silkworms, the spiders. And in order to identify what type of proteins are involved, you have to dissolve the structure. And this can be quite tricky. I mean, some of you might have tried to dissolve a silk fiber, and it's very, well, complicated. You need some very harsh conditions. In that case, you cannot do that with just, increasing the temperature or changing the pH or put on some pressure. The only thing that works are so-called Hydrophobic substances. Substances that interfere with H bonds that are occurring between the amino acids inside the structure. Some typical Hydrophobic substances are urea, you know about that. A stronger one is guanidinium chloride, but these two still don't work on silk. If you work with silk, you need really strong denaturants like lithium bromide or guanidinium thiocyanate. So these are some of the strongest Hydrophobic substances, and you need them at really high concentrations. So we talk about 8 to 10 molar concentrations. So really strong substances to unfold such a molecule. And this is necessary if you would like to understand what is within a fiber, right? And therefore, I would like to have a very brief word on that. So typically, if you work with an enzyme or a simple protein, you just can raise the temperature, like in case of eggs, unfold the proteins just by increasing temperature. They unfold and then they start to aggregate. You can do that by lowering the pH, for instance. You can induce pressure. You have a pressure-induced unfolding. So question is, when you can unfold a molecule with increased temperature, can you also unfold it with lowering the temperature? What do you mean? Can you have a cold denaturation? It's a 50-50 chance. So if you increase the temperature, you play with the entropy. What happens if you decrease the temperature? Yeah. Yeah. So what will happen? Will they unfold? You don't think so? Well, it depends on the temperature, of course. Yes, you can cold denaturate all proteins. The reason is that you have all the hydrophobic side chains packed inside a structure. And what I didn't tell you so far, hydrophobic interactions are extremely temperature dependent. So the lower the temperature, the looser the hydrophobic interaction. So if you can cool down a solution to -10, -15 degrees centigrade, most of the hydrophobic interactions are so loose that they are no longer stabilized. And therefore, they don't care anymore if they interact with the solvent or not. So you denature the proteins. And because the hydrophobic effect is gone at these low temperatures, it does not aggregate. So meaning, if you would, instead of boiling your egg, freeze your egg, you would have also an unfolding of the molecules, but you will not get the egg white because it will not aggregate. The problem is that for most cold denaturations, you have to go down, even below -20 degrees centigrade, and this is, well, somewhat tricky in water. You can work with supercooled solutions, but you have to add a lot of salts, etc., etc. But theoretically, it's no problem to cold denature proteins. Practically, it's complicated. But it would be the smoothest way because just by raising the temperature, you can refold it, which is not the case with high temperature, because if it's aggregated and you lower the temperature, you cannot come back. And with the chaotropic substances, you have to remove the chaotropic substances first before you want to come back. And I will come to this later on, because this is one issue if we work with silks. We denature the fiber with these very strong chaotropic substances, and then we have to remove them again if we want to make a fiber. So this unfolded silks we call also regenerated silk, and you might have heard about that. And then you can make fibers out of regenerated silk, but in case of doing so, you have to remove then the substances, and this is tricky.
But you mean that you decrease temperature enough, and then you increase it, you will have your protein again.
Yeah.
So how can you work with that?
As I said, I mean, the problem is that it's very tricky to get to cold enough temperatures to unfold all the molecules. So therefore, in experimental setups, this doesn't work. I mean, it works in some cases, but in most it doesn't. But just generally, this would work very nicely. And with these few molecules where they've shown it, you just can raise slowly the temperature again, and it folds correctly. So just on its own, without any side reactions. So that's the cleanest way of unfolding and folding a protein. And this is why some people, when they have some disease, whatever, they, let's freeze them, and because they think, well, they can thaw me like in 100 years, and then they can cure my disease. Because this works really. If you avoid, and now this is one major point, if you avoid the formation of ice crystals, because ice crystals will destroy the structures. So this is the major point. You should have no ice formation inside, because that is deadly for the molecules. But it should work, and therefore, maybe these people can be cured in 100, 200 years. Maybe not. Okay. Spider silk. Protein fiber, very strong. Now I would like to have a more closer look about the material. I mean, you're material engineers. If we talk about spider silk, I mean, this is actually just like if we talk about milk, right? But milk can be different. I mean, milk from cows is different to milk from goats, etc. If we talk about spider silk, even from a single spider, it's not a single material, it's a whole set of different types of material. And this is what you can see here. So this is a European garden spider. And that's one of the major spiders we're working with, but we also work with spiders from Asia and South America and Africa. So this spider makes what we call orb webs. So they look like a wheel. And these orb web forming spiders, they actually have the highest repertoire of silks, because the female orb weavers can make up to 7 different types of silk. Different in terms of composition, different in terms of silk proteins. So actually this spider, so this would be the back part, this is this, what I drawn yesterday. That's the silk factory. And for each of these silks, the spider has an individual gland, and not a single one, it always comes in pairs. So this spider has 14 glands, 2 for each of the silks. And now I will introduce these different silks a little bit more closely. Let's start with the web. So first, if you watch a spider making a web, it starts making a frame. And for this, it uses a silk that is produced in the matrix ampulate. So that's the name of the gland. And the reason is, it looks like an ampulla. It looks pretty much like this. Biologists saw that in the microscope. That's the major ampulate, because there's also a minor ampulate, which is just smaller. So it looks like an ampulla, and the silk that comes from that gland is called the major ampulate silk. And this is used for making the frame, it's used for making the radii of that web, and it's also a lifeline. So that's the fiber the spider hangs on. The next silk is called flagelliform silk, because here the gland looks like a flagelle. Don't ask me how flagellae look like. Different type of silk, different type of properties, because while this major ampollate silk is quite tough, it's a very strong silk, the flagelliform silk has to be very elastic, completely different mechanical behavior. Because the flagelliform silk is used as the capture spiral. So this is actually the hunting part, or the prey hunting part of the web. If you just touch that web, you will find it's sticky, it's gluey. But this is not the fiber itself. It's a glue coming from a third gland, from the so-called aggregate gland. It's the aggregate silk. And that is forming the sticky coating on the capture spiral, on the flagelliform silk. Then we have sort of like a supporting silk, which comes from the minor ampollate. So it looks pretty much like the major ampollate, but it's just smaller, the gland. It makes sort of an auxiliary spiral. We have seen such spiders sometimes sitting in the middle of a web, and they're sitting on that sort of hub, and it's also used for reinforcement of the web and stabilizing the web, and sometimes it just comes together with the major ampollate silk. So these two are closely related, also in terms of mechanical properties. And it's very important that the glue will just be put on the capture spiral, but not on the frame and not on this reinforcement fibers. So therefore, the spiders have like at least one chance not sticking to their web. They have other mechanisms to avoid sticking. There's just been a publication a few weeks ago that they also have very specific tips of their toes that they do not stick very well to the glue-like substances. But I mean, in simple terms, they typically just run on the fibers that are not coated in glue, and therefore they don't stick. We have also some kind of cement, which is coming from a gland that's called the piriform. So this is used to attach the web now to substratum. It could be a tree or a wall or whatever. So again, something like a glue. And then the female spiders have also additional silks used for coating their egg cases, the inner egg case, the outer egg case, and so on. So what all of these 7 silks have in common, they're mainly made of silk proteins. But what is different is the type of silk protein, the size, the molecular weight, the amino acid composition, and also the amount of proteins. In a major ampollate silk, we find at least 2 different proteins. In flagelliform silk, we find a single protein. In aggregate silk, we find a whole series of proteins. So not only that the individual silks differ in their properties derived from individual primary structures, they are also sort of blends of different molecules, which are then again different. So such a spider can probably make up to 20, 25 different silk molecules, and they all differ. This is very important to know, because if we talk about silk, we typically talk about major ampollate silk. This is what we know most about. And now, recently, we also get information on the other types of silk, but most is known about major ampollate silk, and everything I will tell you from now on will be about major ampollate silk, because this is what we know most about. So this is what we've seen yesterday, and that's a typical setup of spidrin, a silk spider protein. And this is, of course, then also the typical setup of a major ampollate spidrin. We heard about these globular domains that are structured in solution. And we have this core repeat unit, which is unstructured. We call that intrinsically unstructured. It has no structure, meaning it's well solved in water. And this is due to the case that we really have a lot of amino acids that do interact with water very well. A lot of glycines, serines, prolines that break up structure. And it's the combination of the amino acid sequence or the primary structure that makes this core sequence so well dissolved. So what we have to do actually, if we want to process that, is quite simple. What do you think? What do we have to do? This is well solved. It's here in the gland. We want to make a fiber. So what is the key issue in folding such a protein? And this is what the spider does in the very first glance. I mean, if you have something that's well dissolved in water, what do you have to do? You have to remove the water. And this is what the spider does. And this is what I will tell you tomorrow, how the spider removes the water. So that's actually the major trick the spider uses to make a fiber out of these proteins. It has to remove the water, and it has to recycle the water. Because water is, of course, very, very valuable. So the spider cannot just, lose the water all here in the back. So it has to have a very precise mechanism of removing the water and recycling the water. And this is what I will tell you tomorrow, how this works. But what I will tell you today, and probably we will make a break here after that slide, then you can change the tape, is that we would like to have now spitrins, individual spitrins, out of this whole set of molecules where it's very complicated to find out which protein does what. We would like to get single spitrins for analysis and probably also for applications. Why is it better for analysis? Well, that's clear. If I have a single molecule, I can really dedicate everything I see to the single molecule, and there's nothing else influencing it. Why for applications? Well, if you hear about silk applications, and you heard about silk applications in the past, one big drawback has been that the quality of the silk differs quite a lot. I mean, if you use silk for textiles, there are even technical qualities that differ than good performing from low performing silk. You even can see that if you have a cocoon of silk just by color, for instance. But even with an individual, the quality of a silk differs depending on the day, on the environmental conditions, temperature, humidity, on the diet. And this makes it very hard to get reproducible characteristics to a product, especially if we talk about biomaterials. We need high precision products that perform every day in every patient identical. And this is much easier to do if you have a single component than you have a mixture that you cannot control. And that's the other reason why we are looking into getting spitrins, individual spitrins, not just for analysis, but also for applications. So what are the problems? Problems are that natural proteins are not very well available, at least for spiders. I mean, it's well established for silkworms. You can farm them large scale in India, in China, even in Europe. That has been done for centuries. This works fine, but then you have the fiber, and you have to denature the fiber with our chaotropic substances, and then you have to remove the chaotropic substances, and you have to be sure that everything is removed. You do not want to have one of these substances in your body. That would be no good because they're highly toxic. So this is a big issue. If you want to have the proteins, what you would have to do is you have to dissect the animal and remove the proteins. And there's a company in Germany founded by Fritz Feurath. It's called Spintech. They do that. So they farm silkworms, and instead of just getting the fibers out of that, they just dissect the silkworms and remove the proteins from the gland. They've actually invented a machine that does that. So because if you do that manually, it will take forever. So they have a machine to do that. So that might still work. It does not work for spiders because most spiders are cannibalistic. So if you try to hatch them, they start to eat them. And we made the same mistakes as probably everyone. So my students, they went to a big spider fair and bought spiders and had them all in one box. And they came back like Saturday night and put them in the lab. And on Monday morning, we were like, whoa, we forgot to lose this box because there's just 2 left. And then we just found the remnants of the other spiders. So within 2 days, I mean, out of a number of 20, they went down to 2. So they're really very aggressive, especially if they're in a closed environment. So it's no good to keep spiders in a room. I mean, you can have some of them together, but not many. And this is not enough to get materials, especially then if you have to kill the spiders by dissecting it to get to the proteins. So the natural source is by no means an optimal thing, so that's a big problem. So therefore, people started like 30 years ago to biotechnologically produce the proteins. That would be a way getting the genetic information from the spiders, transform them into any other host, and then just produce the silk proteins there. And you might heard about the goats that make silk in milk, which technically works, but practically fails because in milk you have a lot of other proteins like caseins, and caseins are proteins pretty similar to silk proteins. They're intrinsically unfolded, they behave like silk proteins, and if you try to remove caseins from silk proteins, you fail. They will also co-purify. So what you then have finally is a silk milk fiber, so to say. And this is one of the reasons that that the company stopped that project. Another reason is that you need a lot of goats. I mean, we just calculated that for a production in 1 cubic meter of bioreaction. So this would be a reactor, it would fit in that corner. You need 10,000 goats. Just imagine keeping 10,000 goats, you need area. You need someone who removes, like, you know, the leftovers. You need someone who milks them every day, and so on. Again, not very productive. That was the other reason why the goat project did not work. So what you want to do is to not use big animals, but use small animals or microbes. That would be one thing. Or you can use plants. But with plants, there are other issues. I don't want to go into much detail there. So you want to go into microbes. Best would be bacteria, because then you can have, like, you know, something like this, that size, and you can produce quite a lot of them, billions of bacteria, and they could produce your silk proteins. But now we have a problem, because we have a highly repeated sequence. And simple organisms like bacteria, they don't like repeated sequences, or they think, well, that's not necessary, and they start to chop it down. We call that homolog recombination. So if you introduce a silk gene in a bacteria, like 2 days later, it's roughly about 1/10 of the size. So you don't get the material you want. The other issue is what we call codon usage, and this is what I will explain after the break. Okay. So we finished the first part saying, well, there is a problem in the organization of genes. Highly repetitive sequences. We heard that already, talking about the primer structure, and codon usage. And this is something I would like to expand now a little bit. So this is what we call the way from the genetic information, the DNA, to the protein. First, we have DNA. You all know that. It's a double strand, and in order to transfer that information into a protein, we first have to make a transcript, and that takes place typically in the nucleus of an organism. And actually, a copy is made, which we call messenger RNA, of the DNA, and this is now the copy that is then transferred to the ribosome. So that's the machinery that makes the proteins. So the messenger RNA is recognized by the ribosome. It's running through, and then actually we have here encoded the information for the amino acids, and the amino acids are then just linked together by the polypeptide bond. We heard about that. We start always with the amino terminus, so the first amino acid here would be the starting amino acid, and then it goes on and on and on, and they're added here in the ribosome under the consumption of ATP. So this is an energy-driven processing. And this encoding functions typically on the so-called codon usage. What does that mean? Well, you know that DNA, RNA is made out of 4 different nucleic acids, and they're just chained together. And always 3 of them are called a codon. So you need 3 of these nucleic acids to specify a single amino acid. For instance, here, this is the amino acid tyrosine, and you see here the triplet code AUA. So that would be now here counterparted in the messenger RNA. Of course, this would be UAU. So you have a specific codon for that specific amino acid. Now, if you think about having triplets as a codon, you can imagine that with 3 different, so if you have 4 different nucleic acids and you have 3 of them in a row for encoding an amino acid, you could encode 64 amino acids. But as we learned yesterday, we only have 21. So there is sort of a redundancy in the genetic code, what we call. Of course, we have some of these triplets that determine a stop, which is very necessary, because if not, we will not have a precise ending of a protein. It will just go on forever. So 3 of these triplets are deciphering a stop codon, but then we still have 61 left for 21 amino acids. So this means we do have codons, we do have amino acids for which several codons will work. And we call that the degeneration of the genetic code. So this means that we have several triplets that encode one and the same amino acid. For instance, the amino acid alanine, I think there's 4 different triplets known to encode alanine. And the point now is that the usage of these triplets is different in different organisms. So humans use a different set of codons more often than bacteria. And this is now a very big problem if we talk about silk genes, because spiders and also silkworms have a completely different codon usage as bacteria do have. And what makes a difference? Let's say we have 3 different codes, code 1, 2, 3, and 4. And in spiders, this code 1 accounts for like 80% of the amino acids. Let's say alanine is encoded to 80% by this codon number 1, and just, let's say, 15% by this codon number 2, and then to the lower amount, let's say 2 and 3% by these other codons. And now in bacteria, that's quite different, because bacteria might actually use codon number 3 most often. But there's not that many codons now in our silk gene, means that bacteria have a big problem in encoding these triplets that are most often used in spiders. And this is what I've meant here in problems with the codon usage. So we do have codons in spiders that are very often used, but they're not very common in bacteria. And this is a big issue, and this is what we wanted to overcome with our technologies. So typically, what you would do if you want to biotechnologically produce a protein, you have to get the genetic information of the animal, in this case spiders. So you have to get DNA. There's several techniques for getting DNA libraries, for instance. And as I told you, you can now transfer the DNA to a host organism. And I mentioned already that bacteria are a non-suitable host. You, of course, can transfer the DNA into bacteria, and the bacteria don't care, they don't mind, but they cannot produce proteins out of that DNA because they cannot read the codon usage very efficiently. I always confer to that with like language translation. If you give me a book in Hebrew, I can see there are signs, but I can't read and I can't understand what's written there because I don't understand Hebrew, for instance. And this is the case here. I mean, I can take the book and put it to my bookshelf, but I can't read it. And that's what the bacteria do with the DNA coming from spiders. They just can't work with it. They don't care, but they can't work with it. What you could do, you could use now a host that can deal with that DNA codon usage. For instance, the goats, they could do it. They were good enough, but they produced it in the milk, and I told you about the problems with the milk. Or you can use, for instance, insect cells, and we did that. So if you would like to get more details, you can check this literature here where we published that. So you can use insect cell lines from butterflies, and they're a suitable host because the butterflies have a codon usage that's quite similar to spiders, and you can produce nicely silk proteins, for instance, in insect cell lines. But as I said, not in bacteria. So this would be one route. You can have a host that can deal with it, but you have other problems. I mean, insect cells are not that easy to culture in large scale. I told you about the problems with the goats and so on. So the main goal would always be to come to a simple host that you easily can treat, where it have low costs to get large amounts of molecules. And this is definitely not the case here in insect cell lines. So, and this is just an overview what people tried to do. And I will start with expression of natural genes, meaning a DNA directly taken from the spider transferred to bacteria, such as Escherichia coli, mammalian cells, insect cell lines, bovine and hamster cell lines. These are the insect cells, Spodoptera frugiperda, this is a butterfly, or in Bombyx mori cells. And you see a lot of what we call expression hosts have been tested for quite a few different silk proteins. If you see there, it's major ampulate, major ampulate, major ampulate. So most of the studies have been done with major ampulate silk. There is a study on flagelliform. Well, this is probably our study. And one on tubuliform silk. And as you can see, the protein yields are not very high. Here we're talking about milligram protein purified out of a liter of cell culture. In bacteria, you get 4 milligrams per liter. That's nice, but nothing you can work with. Even the 30 milligrams per liter here in insect cells is not very sufficient. If you talk about application of silk proteins, what would have to stand here is something in the thousands of milligrams per liter. And currently, and I will come to that in a moment, the company I founded 4 years ago have yields in between 4 and 5 grams of purified protein out of a liter. So we're talking about 4,000 to 5,000 milligrams per liter. And this is still not feasible for technical applications. It's good enough for medical applications, but for technical applications, the yield is not high enough because the price would still be too high for getting just simple technical products for that. So for that, you even have to go higher to like 8 to 9 grams per liter, 8 to 9,000 milligrams per liter. This is what you need for applications. So therefore, you see, this is by no means something that will work. You cannot express the natural genes in these organisms. So what do you have to do? Well, this is sort of what we started around 10 years ago. And we thought, well, we know quite a lot about proteins. And we know a little bit about biotechnology, but we found out that you can't just not very efficiently produce the silk genes or express the silk genes in simple biotechnological hosts. So what we decided to do is to analyze the proteins and try to find out what are the key features. So not just to take the DNA as it is, but actually to sort out what we will need from that sequence and to throw away the rest of it. Something like minimizing the information we put into the system to like the lowest level. And then, if you find these protein amino sequences, in terms we're talking about primary structures now, we're identifying primary structures that have a function in silk. And then we take this primary structure, we talk about amino acids, and make a gene out of it. And this is what we call reverse translation. So actually we do something that nature doesn't do. So typically we go from gene to RNA to protein. Now we go back. We take an amino acid sequence and design a gene. But when we design it, of course, we can use now codons to our like. So we can actually now create genes with the codon usage optimized for the host that we are using. So we can make a gene that is perfectly suited for expression in bacteria. So this is what we do. So we take the genetic information and we manipulate it, so to say. We make synthetic DNA with adjusted codon usage, and we remove everything that we think is not necessary. And then we can go to any host because we can do this adjustment to any kind of organism. Can be bacteria, it can be yeast, it can be whatever, plants. And then, of course, we should have a route to our proteins by doing that. And you see that this is not just an idea that we have started and this was it. I mean, there's a whole list of sources, silk sources that have been already successfully produced. So designed genes, mainly in bacteria, some in plants, some in yeast. Yeah, that's pretty much it. And you see now we get much better, well, at least in some cases, much better production yields. Here we see already 1,000 milligrams per liter, 300, 360 milligrams per liter. This is just to show you this works much better than the other approach. Of course, you have the drawback that you have a designed gene. It's not the natural blueprint anymore. It's a mimicry. But that's still fine enough. If you want to have an application afterwards, you don't care if this is the original or if it's a blueprint. If you have a high-performance car, if you think about Formula 1, you don't care if this is the original Mercedes or if it's the McLaren running with a Mercedes engine, but it's winning. You don't care, right? Unless you're running a Ferrari. I mean, that has to be Ferrari. But that's another issue. So you don't care. As long as you win, you don't care what is in there. So what's the principle? Well, I mean, you just take the fiber, and we've seen different schemes of that, and this is just another scheme. So it's a cross-cut across this fiber. Again, you see the setup in the middle. You would have the fibrils. In this cartoon, there's not the fibrils shown, but structural elements. We have beta sheets, the white boxes. These are the beta sheets, the crystallites. There are small crystallites here. The small crystallites come from the polyalanine sequences. There's the larger crystallines here. And the black surrounding matrix, that's the amorphous structure. It's not to scale because the skin here is much thicker than in real. I mean, in real, the skin, which is made of lipids and glycoproteins and so on, is just a few nanometers, while the entire diameter is 3 to 4 microns. So it's just a little bit overdrawn here. But what we're interested in is the structure in the middle. And actually, we have here different types of structures. The amorphous phase, we have the large crystals, the low crystals. And this, of course, was the focus of our research. We wanted to identify, and here in orange, that would be a single silk protein. It would go on and on and on. And we wanted to find out what kind of primary sequence, what kind of amino acids are responsible for the individual structures. And this has not just been done by us. I mean, there's many, many people doing research on that, and they started in the 1950s with X-ray diffraction studies and so on. And there's quite a large database on the structure-function relationship. And it's pretty clear that these small crystals are based on polyalanine sequences. So the crystal sizes are roughly between 2 and 3 nanometers in all dimensions. The larger crystals are typically formed by glycine-alanine runs that are then repeated over time, and there we have like 20 by 70 nanometer crystals, and then of course we have the amorphous phase. So if we just take now one of our model silk proteins, as I told you, we will work with the garden spider, and we take the major ampollate silk of that only. Then we can identify the DNA sequences for the spidrins, and then we can, of course, identify the sequences of the terminal regions, but also of these ensemble repeats, of this repeating sequences in the middle. And what we have to do is now to adjust these sequences. We can put them in some plasmid DNA, so do some molecular biology, have the engineered spidrins, and put that now in bacteria, and they will produce the proteins for us. So this is actually how this reverse translation works. So we get the amino acid sequence. Here's a polyalanine sequence. That would make a crystallite. Here is a typical matrix peptide, glycine, proline, glycine, and then there's 2 glutamines. And you see that is going on and on, GPGQQ, GPGQQ 4 times. This is a typical matrix peptide. This will not make structure. This is amorphous. This is flexible, an elastic structure. So what you do then, you just take this amino acid sequence and back translate that into oligonucleotides. You do that on a computer. And then you just order that DNA strands. So you have tiny little pieces of DNA. And now comes a trick. Now you have to put them together. But if you put them together, and this was one of the major advances that we made, if you put them together, you should avoid linking sequences. What is a linking sequence? Well, typically, if you have, I draw it like a box. You have a DNA like that, and then you have the next DNA. You have to put some linker in between to elongate them, to polymerize them. Polymer chemists would say to polymerize them. And typically you have here restriction sites or sites for restriction enzymes that accommodates this linking of these molecules. This was in former days. We developed a technology where we could avoid that. So actually what we could do, we directly could join the individual DNA molecules. And this is what we call seamless cloning. There's no foreign sequence between the 2 blocks. And this is very important because you should not generate additional amino acids in between these molecules. That will be important then for assembly, etc., etc. And this was one of our inventions, and this was one of our patents that we got 10 years ago, that we invented this seamless genetic engineering where we really could put the DNA molecules together. And then we generate a gene, and of course we have now a nice control over the length of the gene, so we can actually control the molecular weight of the protein we make. We can make smaller ones, we can make bigger ones. And we can add the terminal regions, or we just can leave them out. So therefore, we can really play around a lot with the proteins. And I told you that yesterday. This we used for analyzing the function of the terminal domains, because we can make molecules that have the domains and that lack the domains, and then we see the differences and can directly correlate then the function to the amino acid sequence. And I told you yesterday already that these non-repetitive termini, they are there in all native spidrins, and they have a huge importance in terms of stability in solution and also in terms of processing. Again, sort of as an overview, so we take the structural information or the sequence information from a spider. We have repetitive proteins with oligopeptide motifs that are repeated over time. We have termini. We do this back translation. We make small DNA cassettes with single motifs that we put in a designed vector. Here you see such a cassette in the vector, And here you see already the setup that we used for linking now different cassettes, because we can take a second vector here and just join these two vectors there, have a side for restriction enzymes at the other hand. So actually what you do, we have two pieces. This is piece 1. This is a second vector with piece 2. And what we do is we cut here. No, wrong. We cut here at that side. And also we cut here at that side. So we can join these two directly. We would have some leftovers, which we just cleave out. So we remove that, and we remove that. And then we just get, as seen here, this joint DNA oligos. And we can do that repeated times. And as you can see, we can then multiply these individual domains, multiple cassettes. Also, we can manipulate the order of appearance. We can do first 1, 2, 1, 2, but we can also make 2, 1, 2, 1, or 1, 2, 2, 1, 2, 2, whatever kind of combination, and see how this has impact then on the protein structure and also on the protein function. So this gives us now a large flexibility on what we can do with the molecules. And this was sort of like the basis for all of our research, because this enabled us now to make molecules where we really could investigate the structure and function of our proteins. So this is one of the ladies we're working with, the European garden spider. In Germany, we also call it cross spider. You see the white cross here on the back. I don't know if you have it also in Spain. So the biological name is Araneus diadematus. And these are 2 out of several proteins this spider produces. That's the 2 major proteins of the major ampellet silk. And they're called ADF3 and ADF4. And as you can see by the color coding, the sequences these repetitive cores are made of are quite different here. We have 2 motifs in ADF3, which are repeated over time. And we have 1 motif in ADF4, which is repeated here n times. Then we have termini, carboxy termini. I showed you the structure already yesterday, so that was the structure of this domain here. And unfortunately, we do not have sequence information at the other end. And this is unfortunate, but so far we were unable to identify sequences for this end. So we just have information on the back part. What we did in the beginning, now to make this back translation, making modules, sequence modules and DNA cassettes, and then we can actually make synthetic sequences either with or without the carboxy termini, and we can vary n quite in a large regime. We're somewhat limited by bacteria because bacteria cannot produce really large proteins. Like 300 kilodalton is very hard for bacteria to make, so typically we work with proteins with molecular weights between 50 and 150 kilodalton. So this is sort of our setup, and as you can see, we can now play around with that a lot, with length, chain length, with molecular weight, with contribution of termini. So we can actually investigate the polymer character, which is dependent on the chain length. We can investigate the protein character, which is derived from the terminal regions. And this gives us now a huge flexibility. And this is one of my major take-home messages. Silk proteins behave on one side as a protein with protein structure, like in the termini, but it also behaves like a polymer. So you find features that you typically find in polymers in silk proteins, like dependence on the chain length, on assembly, and so on. And it just depends on how you look at it. You will see, it's a polymer, but it then does not behave completely as a polymer because it's still a protein, but it does not behave like a protein, but it is still a polymer. So you can go back and forth. And this is very important if you deal with silks. So in order to get now real mimics, and you asked yesterday, does the amino terminus also work in the context of the entire molecules? And given the fact that we do not have information on that spider, but given the fact that we have information on other spiders like the black widow, and given the fact that we know, and I told you that yesterday, that we have a high conservation of sequences. So they're 99% identical, even amongst different spiders. What we just did, we transferred the sequence of the black widow, the Lactrodectus hesperus, just to our molecules. I told you problem, no amino terminal sequence available for Araneus diadematus, so we just took that one, and this actually allowed us now to make full-length molecules with both termini, where we could now investigate the individual contributions from both ends and how they impact on processing and so on. I will tell you tomorrow. But until we get there, there's now, of course, the big question, what is going on upon processing? So what I've told you so far is a little bit of molecular biology, and we transfer that into bacteria. And what you typically do in the lab in first glance, if you have these bacteria, you have some shaking flasks and you grow them. And then you induce silk production, because typically you control this production with chemical promoters. So you can turn it on and off. So you turn it on after a while and then you produce the whole thing. But you don't get many bacteria. Like out of such a shaking flask, you get probably 10 grams of bacteria. If you want to get probably a kilogram, you go to what we call a fermenter. So this is made of stainless steel. Here's the biggest one I have at the university, has 30 liters of volume, so we can make 30 liters of bacterial culture. And out of that we get, if we do it right, between 2 and 3 kilograms of bacteria. However, then if you go to larger scales, then you get to 100 liters, and this is the lab of the company that we founded. Or you even go to, this is 1 cubic meter, so 1,000-liter reactor, but this is still the technical stage. If you go to production units, and this is where the company currently is, we have then 120 cubic meters. So you have steel tanks with 120,000 liters volume. And you have not a single one, you have 19 in a row. So you can imagine how many thousand tons of bacteria you can make within 28 hours, roughly. And this actually is now the limit for production, so you can make within a week tons of proteins. Well, literally, because the protein is still inside the bacterium. And inside the bacterium, it's of no use. I told you silk proteins are highly soluble. So there's nothing going on. I mean, people always ask me, oh, does the bacteria make the thread for you, the fibers? No, the bacteria make nothing. They just make the protein. And the protein is soluble. So it's soluble inside the bacteria. Well, sort of soluble, because it's funny. If you check such a bacterium in the electron microscope, so this is the bacterium, and it pretty much looks in the electron microscope like that. So you have high electron density in like that part, and then you have like typical transparency up here. And this high electron density, actually that's silk protein. So 85 to 90% of the bacterial cells are packed with silk protein. And this tiny little bit is the rest of the bacterium that it needs for survival. This is still soluble. So what you have to do is to get that stuff out. Then it has to get clean. That's what we call purification. And there's now, of course, 2 different means how to get proteins purified. One is for technical applications, and the other one is for biomedical applications. Why is that a difference? Well, for technical applications, what you do, you mechanically crack open the cell wall just with shear forces, so everything comes out. And then you separate anything that's unsoluble. In the lab, that's easy. You have a centrifuge. with 100,000s of liters, you cannot have a centrifuge. So you have separators. They look like a big plate. So these are plate separators, or cross-flow tubings, where you can really pump through thousands of liters per hour to separate that quite quickly. So you remove everything that's not soluble. And then you have all the soluble proteins left. And now we use something, what makes silk so famous. It's very well soluble in water, and it does not aggregate. So you can boil the whole thing. So again, now we come to the egg problem. If we typically boil proteins, they unfold. The hydrophobic side chains are exposed, and they aggregate. When they aggregate, they fall out of solution. So if you open an egg and put that into boiling water, what you will see, you get white flocks, and they just sediment. And that happens with the bacterial proteins. So if you boil the whole thing, all the bacterial proteins will unfold and aggregate. The silk proteins are already unfolded, remember? There's not much that can be exposed there besides the terminal regions, but they're very thermally stable. So actually, if you boil the whole thing, everything from E. coli aggregates and separates. And then you just cool it down and just take the supernatant, and there you have the soluble silk. You need then a final step to remove the water, and we do that by addition of salt. So we just precipitate now the silk with salt, and then we have a white powder. And that's pretty much it. And this white powder is freeze-dried, and then you just can store it or can process it, which we will talk about tomorrow. So for technical applications, that's fairly easy. You can do that large scale. For medical applications, that's now a big problem, because there is some stuff in bacteria, especially in the cell wall of bacteria, which is called lipopolysaccharides. So E. coli has a lot of sugars in the cell wall to, harbor and to protect it from other organisms. And these sugars are also inducing immune reactions, inflammations, and so on. If you do such a technical purification, as I told you, you will not get rid of the sugars, because the sugars are also A, very thermal stable, B, very soluble, so they will not precipitate. So you have a contamination. It's just in the 0.01% weight per weight. It's very low concentrated, but it's enough. If you induce that to an animal, it will get an inflammation or an immune response or whatsoever. So for medical application, this does not work. Therefore, you have to go to good manufacturing procedures, GMP production. So you have to do everything in a clean room. You have to remove the sugars, which makes the whole thing much more expensive. So it's different if we talk about technical or biomedical applications, because although here's just the word purification, the way how to purify the proteins is then quite different. If you actually can purify the proteins, then you're all set. Then you can start with processing. Of course, again, the way how to process it will be the same for technical as for biomedical applications, and I will show you examples for that, where we have like one production for wound coverage devices, and the same material can be used in filter bags for vacuum cleaners. Same thing. But for the wound coverage, we have to do it in a clean room, which is more expensive. And for the vacuum bags, we just can do it everywhere, which is cheap. So the ways to process will be the same. However, we would have to follow different demands depending on the application. Okay, so this is pretty much it. Do you have questions about today? Yep.
When we started with protein in the bacteria, I thought that the silk is not in soluble form. It's making a kind of inclusion bags. What's the real thing?
Yeah. So that's the issue. That's a very good point. I mean, this is what I was told 10 years ago when I started. You will not be able to produce soluble silk. When I wrote my first proposal, the reviewer said, you will fail because the silk is not soluble in bacteria. The problem is, the natural genes have quite many additional informations, so to say, that complicate production in bacteria, for instance. So we have here very low protein yield because of, and you're absolutely right, the natural products are not very soluble in bacteria. That's one thing. And the reason why we succeeded is that by using that technique and by making this seamless cloning technique, we really could remove everything that might interfere with the stability. So in simple words, we did sort of a cleaning out of the sequence. So these are the functional sequences, and in between there is always some chunk that connects the stuff. And the chunk is also responsible for unspecific interactions. So what we tried to do, so that was sort of the idea. We remove everything that's non-functional. And as a result, of course, I mean, we wanted that to happen, but we couldn't predict that it will happen. And everyone else that saw it said it will never happen to be soluble. But indeed, having such a seamless linking of functional units makes the molecules highly soluble. Sorry?
Did you patent the sequence? Do you have, I mean, it's for everything, every people know it's sequence? Or
Yeah, it's all published. Yeah, so that was the first study where we published it in biochemistry. And this nicely works. But you're absolutely right. I mean, we thought we were very clever, and it worked very well here. We worked also with flagelliform silk from another spider species, Nephila edulis. And we failed there completely. We did the same thing as we did here, and there it didn't work. That protein is insoluble. It forms inclusion bodies in bacteria. We cannot make large, so therefore we stopped that project. So we cannot generalize that this always will work. In our case, with that spider, it worked. And now there's also other groups. There's a Korean group that could make it work with silk proteins from other Nephila spiders. So there are some groups in the world that, when using their protocol, can make soluble silks. And they're even soluble then after purification. I mean, this is one of the major issues we will see next, tomorrow, in the next lecture, when I talk about making fibers. Because you can only make fibers if you have highly concentrated solutions. We talk about 20% weight per volume. And this is only possible with these molecules because they're so soluble so that you can keep them in the spinning dope for quite a long time, and they will not fall out, they will not aggregate, and so on. So, but this is a feature of the sequence, of the primary structure. But you're right, I mean, it's a big problem.
you remove some sequences,
Yeah.
to have soluble protein?
So, it's the best to see here. So what we did, I mean, you have a sequence like that. And we identified by bioinformatics and modeling that this would be the sequence that is responsible for making the crystals, and this is the sequence for making the matrix. So this would be the crystal, and here would be the matrix, and then a crystal again. But you see there is something in between, and we just left that out and said, well, there is no obvious function for it, remove it. This also makes then the design of the genes much easier because these chunk in between, that differs between the individual blocks. So you would have to make many, many adjustments individually. Here we just actually design one block for each, and then we just multimerize it. As I said, it's a simplification of the process, or a simplification of the materials, down to like the basic needs that we think, well, that we thought in the beginning would be necessary for the function and also for the processing. And that's a big issue. In other hands, if all the proteins go in inclusion bodies, that would also be okay. Then, in large scale, you also can deal with inclusion bodies. That also works fine. But what is a bad situation is if you have partial inclusion bodies and partial soluble protein. Because then industry cannot mean, either it just purifies the soluble stuff or the insoluble stuff, and you have to throw away a lot of proteins. And this is what industry doesn't like. So that's very important. If
you actually have all insoluble form.
No, we have soluble form. Yeah. At least for the molecules we're working in large scale. As I said, we had some flagelliform silks in insoluble form. We stopped the project. Further questions? No questions. Okay, so tomorrow I will show you how to spin fibers artificially. Well, nature, how nature spins fibers, how the spider does. And major issue, removing water. And I will show you how we do that in the lab, biomimetic spinning, but also then other spinning techniques, and how we come towards applications. And that will be then the final 2 lectures. I show you then applications in drug delivery and tissue engineering. Thank you very much for your attention.